Okay, so welcome everyone to this Population Dynamics virtual seminar. The speaker of today is Fiona Bate from University of York. She will present something regarding how to use proteome structure to predict the lag phase in microbial populations. So some words regarding the speaker. She got a PhD in 2004 at the University of Birmingham. And then she worked at the Harvard Science and Innovation Campus in Oxfordshire. And now she is a Daphne Jackson Research Fellow at the University of York. And she's interested in mathematical biology and chemistry, and she worked also in optimizing biofuel production in spatial models of wastewater treatment. And today I think she will tell us something more on a new story, or at least what I got is that one. And so, Fiona, actually the stage is yours. I will stop sharing my screen. And just for the audience, they can ask questions during the talk. If you need, you can also put the question in the chat. I will read it aloud. And the talk will be recorded up to the question time. So if you don't like it, just point it out. And yeah, Fiona, the stage is yours. Thank you.
Thank you very much. Just going to share my screen and then I can start. Okay. Hopefully you can now see my slides. Yep. Okay. Excellent. Thank you for inviting me to come and talk to you today. Thank you to Marco for the introduction. I'd just like to add in my career profile, it didn't actually show there was a very long career break. So in between working in Oxfordshire and getting the Daphne Jackson Fellowship in York, I actually had an 11-year career break. So I'm just getting back into science now through the Daphne Jackson Fellowship. Okay, so today I'm going to talk about using proteome structure to predict the lag phase in microbial populations. I'm going to talk you through a mathematical model I've been developing over the past couple of years. Okay. So the motivation behind this work is wastewater. Wastewater is basically used water from baths and showers and toilets and washing up and things. All the water that goes down the drain in your house and ends up in the sewers. If this water gets into rivers and the sea untreated, then this can cause really bad problems for the aquatic life because all the organic matter in the wastewater gets broken down, which uses a lot of oxygen that's dissolved in the water. So you get dead zones. You can't really sustain aquatic life. So to get around this problem, the water goes to a wastewater treatment plant. This picture here is of the Nostrop wastewater treatment plant, which is in Leeds, which is just down the road from where I am in York. Okay, so at the wastewater treatment plant, the organic matter in the wastewater gets broken down under controlled conditions. It produces something called sewage sludge. And what I'm really interested in is the anaerobic digestion of this sewage sludge. So that is a process in the wastewater treatment plant, which happens in this region here. I'll just zoom in. So hopefully, if I just put my pointer on, these 4 tanks here, there are 4 anaerobic digester tanks. And inside these digesters, the sewage sludge gets broken down by lots of anaerobic microbes and produces biogas, which gets collected in this sort of bubble here. Biogas can be used as a fuel instead of natural gas. It's a green energy. Okay, so I've just got a little schematic of anaerobic digester. What happens is the untreated sludge goes in at the bottom of the digester. The microbes inside the digester break down the organic matter and you get treated sludge, which is much cleaner than the untreated sludge, and you get biogas as well. So the process of anaerobic digestion, you've got 4 main stages. I'll just go through them now. First of all, you start with your complex organic material, so carbohydrates, proteins and lipids in the untreated sludge. These get broken down by hydrolysis into basic monomers, monosaccharides like glucose, amino acids and long-chain fatty acids. The next 2 processes, acidogenesis and acetogenesis, break down these monomers into fatty acids, acetate, hydrogen carbon dioxide. The final process in the anaerobic digestion is methanogenesis, where the acetate gets broken down into methane. All the hydrogen and carbon dioxide react together to form methane. So the microorganisms in the anaerobic digester, they break down the complex organic material into the simpler compounds, using some of the carbon and energy from this breakdown to grow. The populations inside the digester, so all the microorganisms, they're syntrophic, they're all working together. So like the hydrolyzers, the products they produce are used by another group, the acetogens, these are acidogens. So they're the substrates for those other groups, so they all work together. Changes in just one group, I should probably point out there's 4 main groups, the hydrolyzers, the acidogens, the acetogens and the methanogens. And if one of these groups, the population either gets really large or small, then it can affect the whole digester process, so the performance of the digester can be affected. The amount of biogas you get will be affected as well. And populations, an effective population will take a long time to establish inside a digester. Any changes will take a long time for the population to establish itself as effective. So if there's a change, such as if you need to restart a plant, or if the sludge coming into the digester, if the quality of that changes, then the population will take a long time to adjust. So your process will be affected and not be as efficient as it should be for a long time. As I've said, the microbial population requires time to adjust to these changes. Restarting a plant is slow. It can take a couple of weeks. And if you're sitting around in the water company, you want everything to be running efficiently. You don't want to have to take 2 weeks out of your schedule to restart a plant. So a better understanding of how the microorganisms inside the digester adapt to the new environments, it could potentially save the water companies time and money. Okay, so what are we doing about this? In York, we've got a lab in the biology department, a System 60 it's called. It's got 60 small digesters. Hopefully you can see on the picture all these black cylinders here are the digesters. There's a close-up of one here. So the sludge in this case gets fed in at the top, and this is the overflow where the treated sludge comes out, and the gas gets collected up at the top here. So the fact that we've got 60 of these digesters means that we can do replicated experiments, quite a lot of experiments at the same time, to investigate the community, the anaerobic community, and how it is changed by things like which microorganisms are in there, and the food that we're giving it, and temperature and pH and things like that. What I am focusing on is the response of the community to an injection of new growth media once following a period of starvation. So the sludge that we use in this lab comes directly from the wastewater treatment plant. So the people in the biology department drive to the wastewater treatment plant and collect big tubs of sludge, bring it back. And when it comes into the lab, it sits for about 2 weeks until it's basically inactive. So we no longer see gas being produced or anything like that. The microbes are just dormant. And once it's reached this stage, fresh growth material is injected into the digesters. We've been looking at injecting fatty acids, things like octanoate and hexanoate and things like that, and looking at the response of the community to an injection of media after it's been starved for a couple of weeks. Okay, so now I'm going to talk through the model that I have formulated to go along with the experimental work that we've been carrying out. So we'll go back to basics. When a microorganism is subject to a change in conditions, it's got to adapt. So to grow on a substrate, it expresses a set of proteins, which is called the proteome. When the conditions change, the proteome needs to adapt. You need a new set of proteins to enable growth on a new substrate. And while the microorganism is adapting, its growth slows or even pauses, and this is called the lag phase. Okay, so I'm just going to go into a bit of information about the proteome now. We're using a model of proteome partitioning, which was first published, I think, by Scott et al. In 2010 in a paper in Science. In their model, the proteome is split into different sectors, and each of these sectors is composed of proteins whose expression levels show similar growth rate dependencies. And there's a global regulation of gene expression here. So all the proteins in a given group will change their expression levels in the same way when the growth conditions change. So it wouldn't just be one enzyme that gets upregulated, it would be the whole of the sector. So I'll just describe what each of these sectors is. The C sector here is made up of catabolic enzymes that break down substrate into carbon precursors in the cell. The A sector is anabolic proteins that take the carbon precursors and build them into amino acids. The R sector is ribosomes, which take the amino acids and produce proteins that are needed for growth. The sector here, which is sector U, is uninduced proteins, so they're not in the catabolic, anabolic or ribosome sector, but the size of that sector is related to the size of the ribosomal sector. So all four of these sectors here, the size will change depending on the growth conditions. There's one more sector that we're looking at, which is sector Q, and these proteins are expressed at the same levels independent of the growth rate. The mass fraction of this sector is constant. Okay. So as I've said, the four sectors here depend on the growth rate of your microorganism. This sector is constant. So some substrates are better than others for a microorganism, by which I mean that they'll give a higher growth rate and faster doubling time. When you've got a better substrate, then the sizes of these sectors will change. So hopefully you can see the C sector, in this case, for a better substrate, you need less catabolic enzymes. So this sector is smaller, and these three other sectors, their size increases. So on the other hand, if you have a worse substrate, which will give you a lower growth rate, you need more catabolic enzymes, and the other three sectors here will get smaller. So the total amount, oh sorry, before I say that, sector U, as I said previously, is related to sector R in the proteome partitioning model, we just take a constant dependent. So this epsilon is just a constant. I've taken the value from this paper by Yu et al, which is 0.3. So whatever the size of the ribosome mass fraction is, the size of this sector is given by 0.3 times it. And in total, the amount of protein is fixed. So we get an expression for the mass fractions of the 3 sectors of the proteome that we're interested in, the ribosomes, the catabolic enzymes, and the anabolic enzymes. If you add them up, then you get a constant. This phi max is a constant, which is just equal to 1 minus the mass fraction of the Q sector. So this is the constraint on how much of each, how the mass fractions of each sector vary. If you increase one, then the others are going to decrease, just because the protein amount is fixed in the cell. Okay, so the next...
sorry, Fiona, we have a question in the chat. Vinod is asking, how common is to have some protein partition, same protein partition in different bacteria? So I think it refers more to the way in which people are intending this kind of modeling, protein partitioning in different bacteria.
Okay, I think most of the studies that have gone on, they've looked at E. coli, because everyone seems to study E. coli. So the papers that I've looked at for the proteome partitioning models, the data is for E. coli. I think it can be more general though for different bacteria. There's always going to be a fixed amount of protein in each of the microorganism cells. So if you increase the number of ribosomes, say, then you're going to have to decrease the number of the catabolic enzymes. I think it's generalizable to microorganisms, although from the literature at the moment, most people have studied E. coli. Does that answer?
Yeah. Oh, sorry. Yeah, I think that was the question actually.
Okay.
Yeah, I think so. Thanks.
Okay, right. So, okay, so this is just a schematic of the core mechanisms that we represent in our model. External substrates are brought in to the cell and are broken down by the catabolic enzymes into precursors. These are then built up by anabolic enzymes into amino acids, and then the ribosomes build proteins. How much of each different protein is produced is decided via regulation functions in the model, which I will talk about later on. So we adopt the flux controlled regulation model formalism of Erickson et al, which I don't know whether you can see, is in a paper here, which was in Nature in 2017. We split the so there's a flux flow, sorry, through the whole process from the substrate coming into the proteins being built. There's fluxes happening through here, which we split into 3. First of all, the carbon influx, where the substrate comes in and gets broken down, and you get a pool of carbon precursors formed. Secondly, we have an amino acid synthesis flux, where the precursors get built up by the anabolic proteins to produce amino acids. And the third flux is the protein synthesis flux, where the ribosomes use the amino acids and produce proteins. So in our model, there are 3 sectors of proteins that we are looking for, and each of these will be made in as a different fraction of the total amount of protein, depending on the growth conditions. I think biologically, the choice of which protein to make is regulated in response to messenger molecules such as PP, GPP, and cyclic AMP. And they're determined by the size of the amino acid pool. Then if there is a lack of amino acids, then PP, GPP will inhibit the production of ribosomes. And in a similar way, the cyclic AMP will regulate which proteins are made and in which quantities, depending on the growth conditions. Okay, so the total protein synthesis flux, which is JR, will be related to the growth rate of the microorganism. In our model, we assume that it's directly proportional to the protein produced. And this is based on experimental evidence that the total protein mass as a fraction of the total biomass is relatively constant. So as you increase protein mass, the biomass increases at the same rate. So we take the growth rate to be proportional to the protein synthesis flux. So on a timescale that we are interested in, which is the timescale of growth of the microorganism, which is around about 30 minutes to an hour doubling time, we assume that these fluxes inside the cell are all balanced. So any changes, they're going to happen of a much shorter timescale than an hour. So we can write that the protein synthesis flux is just as constant times by the amino acid synthesis flux. And similarly, the amino acid synthesis flux is a constant times by the carbon influx. So what do these fluxes actually depend on? Now, the rate of protein synthesis is going to depend on the concentration of ribosomes and the average translation rate, which we call sigma A. And we just take a simple expression here. We just multiply the concentrations together. So we're not looking at anything complicated. It's just one thing times by the other. It's a first order rate. And similarly, we say that the amino acid synthesis rate just depends on how many anabolic proteins you have, which is phi A, times by sigma C, which is the amino acid synthesis rate. Now, the carbon influx rate is slightly more complicated. Put the expression on here. I'll go through each bit, so hopefully you will understand where it's come from. Now, the carbon influx rate will depend on the external substrate concentrations, and it will be dependent on the substrate uptake rate. And we've based the substrate uptake rate on Michaelis-Menten kinetics for the rate of the substrate uptake reaction. So Michaelis-Menten kinetics basically says that the reaction rate depends on substrate concentration, which is this Sj, in the following way. You might have seen it more commonly written like this, Sj divided by K, Sj plus Sj, where this is an affinity constant for the substrate, but it's exactly the same as this, just divided through by Ks. Now for multiple...
Sorry Fiona, just a question. I mean, maybe two, because there was also one before. In the slides before, the rates were at steady state, were given as proportional but not equal, right? If you go on the slides before. Yeah, here.
I got a bit lost here.
JR proportional to JA, they were not equal. Why is alpha?
Here?
Yeah.
What's alpha?
This is a constant. So it's just saying that the flux, the protein synthesis flux is just a constant times by the amino acid synthesis flux. And this is just it's going from amino acid flux to protein
is it like
synthesis.
how many amino
Precursors can actually build up a few amino acids, yeah, so it's a conversion factor, yeah, that's fine, yeah, now we got. And in the next slides, for regarding the choice of the function for the substrate, I mean,
Yep.
Mason works only for independent substrate, like I don't know, substrate that can be taken up at the same time without interfering on each other, right? Because there are some substrates that can suppress or creating some deoxy shift or other things. These
Yeah, I'm just going to get onto that.
Okay. Yeah.
Yeah.
Thanks.
Okay.
Thank you.
So, okay. So in the Michaelis-Menten kinetics for one substrate, you get this form of dependence on the substrate concentration. When you have multiple substrates, so if you have one enzyme that catalyzes the reaction of two substrates, then you get the reaction rate depends on all of the substrates that can be catalyzed by that enzyme. So in our model, we've got the C sector as a whole is used to catalyze all the reactions. So we want to, it's basically being shared between all the substrates. So that's why we've got this sum in the bottom here. We've based it on the multiple substrate kinetics. Okay, so the reaction rate doesn't just depend on the substrate concentration, it also will depend on how much enzyme you've got, which because the whole of the C sector is regulated as a whole, the amount of enzyme is directly proportional to the mass fraction of the C sector. Now we've got this phi star Cj. This is the value of the mass fraction of the C sector when the uptake rate is maximum. So when the uptake rate is K max, phi Cj will be equal to phi C, sorry, will be equal to phi C star J. This term will also tend to 1 because you're essentially looking at the case when Sj is really large. So the reaction rate at this point will be K max J. So just looking between what we have here and what we have for the carbon influx rate, we've got this YCJ, which is the yield of carbon precursors. So that's just how many carbon precursors you get per. So it's the carbon uptake rate, the substrate uptake rate is this. So the yield of the carbon, sorry, this is the substrate uptake rate. So the carbon influx rate will be the yield of the carbon precursors. So how many carbon precursors you get for this substrate times by the substrate rate here. Okay. I need to point out at this point that this is not constant. In our model, this yield will change. So just bear that in mind, it's not a constant. The only thing else to note in the expression is that we've got these functions e to j. And this relates to the question I was just asked about whether all the substrates will be used at the same time. So this function e to j depends on which substrates are present. It takes a value between 0 and 1. If a substrate will be consumed no matter what else is there, then e to j will just be equal to 1. But if the substrate consumption depends on which of the substrates are present, so the case of glucose lactose dioxy, then lactose is not going to be consumed when glucose is present. Then we need a form of this function that will be 0 when you've got glucose present, but then will be 1 when there is no glucose present. So for glucose lactose dioxy, we can choose a form of the function like this. And you can see that when you've got large glucose concentration, eta lactose is essentially 0. And when this tends to 0, this tends to 1. Okay. So these functions eta appear in front of the substrate concentrations just to determine whether or not at a certain time that the substrate is being consumed. Okay, so now we want to know how a change in the substrate quality is going to affect the microorganism. So that's the main aim of this model. So say we have an abrupt upshift in the substrate quality, we've suddenly got the substrate that is going to give you a higher growth rate. What is going to happen? So I mentioned before that we were basing the flux regulation on a model of Erickson et al. This is how it's dealt with in their paper, and I'll then explain what the problem with this is and how we've solved that. Okay, so an abrupt upshift in the substrate quality will lead to this carbon influx here increasing suddenly. And because of flux balance, that also means that JA will increase suddenly, and JR as well. So we know that JA is equal to the amino acid synthesis flux times by the mass fraction of the anabolic proteins. Now phi A cannot increase abruptly because the protein synthesis rate is directly proportional to the growth rate. And we know that the growth rate does not abruptly increase when you change the substrate. So the only other way to increase JA suddenly is to increase the amino acid synthesis flux here. But this can lead to unrealistically high values of sigma C. So how are we going to deal with this? So we have noted that sigma C depends on how many carbon precursors you have in the model. So how fast you can make amino acids will depend on how many precursors you've got. How many precursors you've got is going to be limited by the cell's capacity. The cell's a finite size. It can only store a finite amount of stuff in it. So you can only have a finite amount of precursors in this cell. So therefore, sigma C, which depends on how many precursors you've got, must be limited. You can't just keep increasing it and keep increasing it in order to balance flux. So if that's the case, so we can't just keep increasing phi sigma C, then there has to be something that breaks further up the chain here. So what we said, to maintain flux balance, we limit the very first step here. We limit the carbon entering the growth pathway via JC. So this is achieved by allowing the yield of carbon precursors to change. So when you've got an abrupt upshift in the quality of the substrate, then not all of this new substrate will enter the growth pathway. This just means more will be released as product instead of going into growth of the microorganism. So we're not losing carbon anywhere. We've still got a mass balance, still conserving mass, but just the mass is not going into biomass growth, it's going into products instead. We don't explicitly model products in this model, but it could be added. Just wanted to say that we're not violating mass balance here. Okay, so yeah, okay. So we're saying that we've got a finite precursor pool size because of the finite size of the cell. So we denote the combined size of all our precursor pools. So in our case, we've got carbon precursors and amino acids. We say this combined size is P, and the maximum value of P that can be maintained in the cell is capital K. And what we want to do is limit the carbon flux entering the growth pathway, JC, so that JC tends to 0 as P tends towards K. This is analogous to population dynamics where you've got a carrying capacity. So in that case, K would be the maximum population size that can be sustained in a given environment. And the growth rate would then be limited to this carrying capacity. Here, what we're going to do is limit the growth and the flux, the carbon flux, so that JC tends to 0 as P tends to K. And as I've said, we do this by allowing the yield of carbon precursors to change. This is our horrible expression for the carbon influx, and this is the yield of the carbon precursors. So what we do is we write this as a constant YCJ0, and we times this by K minus P over K. So that as P approaches K, the yield tends to 0. And when P is 0, we've just got this constant YCJ0. Putting that back into our expression, we just have this extra term. Okay, so how do we now get a closed model out of this? We've introduced the precursor pool size in this K, and from experiments, we're not going to get these numbers popping out. So how are we going to get a nice model out of this? What we really want is an expression for P in terms of substrate concentrations and mass fractions, which are things that we can actually measure. So we can do this by going back and looking at the flux balance. So here are the equations for flux balance. And from the previous slide, we know that we've got this JC in terms of the total amount of precursors. And if we split this up into PC and PA, whereas this is the carbon precursors, PC, and PA is the amino acids. We've split it up because the other fluxes only depend on one of these things. So the amino acid synthesis flux depends on how many carbon precursors you've had, and the protein synthesis flux depends on how many amino acids you have. Okay, so if we now put these expressions into our two equations, we end up with two equations in two unknowns, PA and PC, which can be solved. Which means that we can get an expression for PA and PC in terms of the other terms in the equation, which is the substrate concentrations and the mass fractions. Okay, which is what we needed. And from these expressions, which I haven't written out fully because they're quite horrible, but they're in the paper if you would like to actually have a look, we can get an expression for the growth rate, because the growth rate is directly proportional to the protein synthesis flux. So this is what we come out with. Fn, these depend on the substrate concentrations, and they look similar to the protein synthesis flux equation, but I've taken out the dependence on the mass fraction. So the mass fractions all appear, they're on their own, they're not hidden inside another function. So Fn only depends on substrate concentration and not on any mass fractions. Okay. These terms here, sigma C max and sigma A max, are the maximum amino acid synthesis rate and the maximum protein synthesis rate, or the translation rate, sorry. So these are constants. Fn depend only on the substrate concentrations, and the others are the mass fractions of the three different sectors. So what we need to note here is that the growth rate is not directly proportional to substrate uptake rate. When a microorganism is growing during the lag phase, the growth rate is much less than the substrate uptake rate. So the substrate gets taken into the cell, but you don't see the growth rate which you would expect from looking at the log scale growth. When you're growing exponentially, the growth rate and the substrate uptake rate are directly proportional to each other. But in the lag phase, sorry, they are not. They don't have the same proportionality. So we were looking for that in our equations, and we've got that. Fn is proportional to the substrate uptake rate, but mu, the functional form, it's not directly proportional to it. Also, for any of these sectors, if we have a very small amount of protein in each sector, then that will limit the growth rate. So say phi r was really small, and these two terms on the bottom would essentially disappear, and what you'd end up with is the phi a, phi c would cancel out, and you just get the growth rate depending on phi r. So the growth rate would be small because phi r was small. And similarly for the other two sectors. Okay, so I've gone through that. Another crucial point is that when you have a fixed substrate concentration, the form of this equation means that there's a unique maximum value of the growth rate at specific values of the mass fractions. We hypothesize that during the log phase, cells will grow at this optimal growth rate. And by doing this, we can find the values of these constants, which we don't know, in terms of experimentally measured parameters like the biomass yield and the uptake rate, which can be directly measured from experiments. Okay, so now we've got an expression for the growth rate and the substrate uptake rate, but we need to know how the mass fractions of the proteins change. So go on to looking at allocating the protein synthesis flux. So the total amount of protein is proportional to the growth rate, but how much of each sector is produced, that will just be a fraction of the total protein produced. I mentioned briefly earlier that inside the cells, the allocation of protein synthesis is regulated via signaling molecules such as PPGPP and cyclic AMP, where PPGPP will inhibit ribosome synthesis if there's a shortage of amino acids, and CAMP will regulate metabolism through coordination of gene expression. And both of these molecules will respond to changes in the sizes of precursor and amino acid pools, adjusting the protein levels until they've got optimal pool sizes. Now in the model, the variations in the pool sizes will manifest as changes to the growth rate, as we've directly put in precursor pool sizes into our derivation of the growth rate. So it follows that an optimal precursor pool size will give you an optimal growth rate. So we allocate the protein synthesis flux using a method that will optimize the growth rate. So we change the levels of our proteins until the growth rate is maximal. Okay. So the easy bit first, sector Q is always produced as a constant fraction of the total protein. We don't need to worry about that. The other sectors, we introduce regulation functions chi-R and chi-C to represent the fraction of the total amount of protein that belongs to each sector. And we define these regulation functions just using a mathematical a mathematical result, just standard calculus. The greatest increase in a function is in the direction given by the gradient of the function at that point. So we want to optimize the growth rate here. So we need to find the gradient of the growth rate and then increase in the direction of the gradient. That's where this d mu phi r comes from, and d mu phi c. This parameter c is just there to make sure that the regulation functions are always positive. In our model, we can't recycle or destroy protein. It's just to simplify things. I won't go into exactly what these are. If you want to know, then they are in the paper that I will give a link to at the end. Okay. So the other sector, we don't need to calculate explicitly because we can just get it from the constraint, which we got from the proteome partitioning model at the beginning. Right, okay, so now we have the closed model. So this model describes the time evolution of biomass growth and substrate uptake and gene expression through these equations here. Just need to note that mu, it looks very simple in here, but actually it's given by this expression here. It depends on substrate concentrations and mass fractions, where the mass fraction of the A sector is just given by the constraint here. Okay, so things to note. We've explicitly included lots of substrates in our model so that a complex system like anaerobic digestion can be described where we've got lots of substrates. And although I've just derived the equations for one species, one microorganism, this X, we can extend the model quite easily to model many more species or functional guilds, groups of species that all do the same thing. Again, which we need if we want to be able to model anaerobic digestion. So we can extend the model with multiple microorganisms, so we can investigate competition between populations. So the growth rate is not proportional to substrate uptake rate, which means that you can model the slower growth which occurs during a lag phase. And all of the phases of growth emerge from the structure of the proteome in this model. We don't have to switch things on and off, changing growth rates at a given time. It's just comes from the structure of the proteome and the substrate concentrations. Finally, we have not made explicit the exact mechanism underlying inhibition of substrate uptake. So we've put these functions eta in, but we haven't said exactly what mechanism we're modeling here. So it's flexible. We can apply it to different processes, not just to dioxic growth. Okay, so before I've started to look at the complex AD system, the anaerobic digestion system, we wanted to see whether the model would actually work on a simpler system. So we looked at the particular case of glucose lactose dioxy of E. coli, as there was data, and it's quite a simple model, but it's relevant to our approach because the data shows an initial lag phase and a dioxic lag phase. So it's showing the lag. So if the model can predict this lag, then it's doing what we want it to do. Okay, so we had a recent data set describing the dioxic growth of E. coli. Experiments were carried out by my colleague, Mickey Amakan, who is in the Chong lab at York. So I'm sure you probably know what dioxic growth is, but I'll go through anyway. When two sugars can be metabolized, then the preferred sugar is consumed first, and then only when that has been consumed, the microorganism switches to the second sugar. And between the phases of growth on the different sugars, you get a lag phase. Okay, so hopefully you can see in the data, there's a dioxic lag here. You can see between the two vertical lines, the growth has paused. There's a slowdown there. Okay, in this experiment, initially the E. coli was grown on a rich media, and then at time 0, it was taken from the rich media, transferred to a minimal media, which just had glucose and lactose on it. So there's a lag at the beginning, which is due to the change from the rich media to the minimal media. And then there's a lag in the middle, which is the dioxic lag from switching from lactose to glucose. The blue curve here is the glucose concentration, the red is the lactose. You can see that the lactose doesn't start to be consumed until all the glucose is gone. Okay, so that's the data we have. Now I'll show you the model predictions. These are the solid lines on this plot here. So we've managed to capture the very slow initial growth here, even though the substrate's been taken up. And this very slow initial growth is due to the proteins being at non-optimal levels for growth on minimal media. When growing on the rich media, things like amino acids are in the media and can be brought directly into the cell, so they don't need to be produced in the cell. And we hypothesize that this means that there's a low level of the anabolic proteins in the cell at time 0 when you switch to the minimal media, which means you can't grow fast because you don't have the anabolic proteins that you need. So that's why we have such a slow growth rate here. The lag here is just due to the fact that the protein mass fractions are not at the right levels for lactose consumption once the glucose is gone. Okay, so hopefully you can see that all the principal features of the growth curve, the black growth curve here, are captured by our model with only minimal fitting. We did have to fit a few parameters, but most of the parameters for the model were just taken straight from the literature. Okay, how long have I got? Not too long. Okay, just quickly, I will share. When we had the parameterized model, we just wanted to see, in a very simple way, competition between two strains of E. coli. So we just extended the model to look at two different strains, one that exhibited dioxy, as in the experiment, and another theoretical strain that does not exhibit dioxic growth. So it will just consume glucose and lactose at the same time. So the plot here, we've got initial lactose concentration on the x-axis, initial glucose concentration here. And we're plotting the final biomass of the two strains, the ratio. So X1 is the dioxic strain, X3 is the non-dioxic strain. Where we've got this line 1, that's where the two strains have the same final biomass. Neither one outcompetes the other. In this region here, the strain exhibiting dioxy has a higher final biomass. So in the case where you've got higher initial sugar concentrations, the dioxic strain will grow more. In the region down here, where X1 over X3 is less than 1, so X3 has a higher final biomass. The strain that doesn't exhibit dioxy actually will have a higher final biomass. So in the case where you've got scarce resources, you want a population that can outgrow its competitors is going to have an advantage. So where we're looking in this case at two strains with the same initial biomass, so the better strain is going to be the one that has the higher final biomass concentration. So these results show that where you've got high sugar concentrations, you should grow dioxically, then you'll outcompete your competitors. Where there's not very much sugar at all, then you should just eat everything all at once, and that's the way to outcompete your competitors. So these results agree qualitatively with experimental evidence. There's a paper by Spencer showing that bacteria with a long dioxide lag will be better in a stable environment. So in this case, a stable environment is this region where you've got higher sugar concentrations, because the longer you've got high sugar concentrations, it means the longer before the conditions will change. And there's also a paper about yeast where we've got the same agreement, where you've got a stable condition, you should have a long dioxide lag, and where the conditions change frequently, having a short or no dioxide lag is better. Okay, so I've presented my model today, which describes the time evolution of biomass growth and substrate concentration and gene expression during growth shifts, up shifts and down shifts we can model. The model includes proteome partitioning, flux control regulation, and we optimally allocate protein synthesis to optimize the growth rate. So we balance all the fluxes in the model, and everything's determined by the sizes of pools of central precursors as to how you balance the fluxes and which proteins are generated. Okay, so the phases of growth come out of the structure of the proteome directly, and we can capture a non-simple relationship between substrate uptake rate and growth rate, which we see during lag phase in experiments. In particular, we can model the lag phase and dioxide growth of E. coli. We've shown that with the match to experimental data. Okay, so currently I'm working on a model of industrial scale anaerobic digesters, and I'm using this model to describe a simplified AD system, concentrating on fatty acid degradation, so octanoate, hexanoate, butyrate acetate, going to methane. That's important in anaerobic digestion because a buildup of acid can affect biomethane production. So the model I'm working on at the moment includes multiple substrates, so the fatty acids, methane, hydrogen, and multiple species, or in this case, functional groups, so acetogens and methanogens, and you've got to have medium chain fatty acid degradators and short chain fatty acid degraders. So there's multiple functional groups, multiple substrates, but we can do that using this model. And hopefully we can also include spatial effects further down the line. So how the flow in the digester affects everything. Okay, so in conclusion, we've got a novel way to predict the lag phase when biomass is subject to a changing environment. This is an important issue in wastewater treatment, but it's also significant in the wider context of studying bacterial growth. Okay, I'd just like to thank my collaborators and the Department of Maths at York. I work with Martin Bees and Mitch Pushkin, and in the Department of Biology, James Chong, Mickey Amakan and Luna Pulford. If you would like to know more, this QR code should link you to the paper that I have written on this. Oh, and also, I should say, my sponsors, the University of York, the Daphne Jackson Trust, and the BBSRC. Thank you very much.
Thank you, Fiona, for the nice talk and great explanation, actually, all through the math. So we'll stop the recording of the moment.